welcome. And today I'm really honored to have the privilege of introducing my friend and colleague, Professor Sabine Kastner, the 2023 recipient of the George A. Miller Prize in Cognitive Neuroscience. Given that Professor Miller spent the brunt of his career at Princeton, I am sure he would be particularly delighted that another Princeton cognitive neuroscientist is the current awardee of the prize in his honor. I think there are three important buckets in the life of a scientist. One, of course, is scientific output aimed at expanding our knowledge. Another is one's relationship to family, colleagues, and friends. And the third is contribution to humanity. All of us work to achieve scientific success. Many, if not most of us, are wonderful colleagues and true friends, and a few of us take the time to advance not only science but humanity. Sabine is unique in that she excels in all three domains. Given the time constraints of an introduction and the desire of all of us to hear from Sabine, I will briefly outline her remarkable contributions to the field of cognitive neuroscience. First, her scientific pedigree. I did not know this, but Sabine started with a BA in history and philosophy. This is very interesting. Explain some of your unique behaviors. I didn't know. Anyway, she then proceeded to do a big switch and got an MD in Heinrich Heine University in Germany, and then moved on and got a PhD in single unit neurophysiology, again in Germany. And then she made two important decisions, I think, which really influenced the rest of her career. One, she did a postdoc in monkey physiology and really learned elegant single unit recording. After she was finished with that, she then went on to another postdoc at the NIMH in DC to learn basically neuroimaging. So now you have a scientist who knows monkey physiology and human neuroimaging. These last two postdocs set the stage for her ability to perform groundbreaking research in both monkey and human physiology, as I will discuss later. Sabine joined the faculty at Princeton in 2013 and rapidly advanced to full professor of psychology and neuroscience. Notably, she has served as director of their imaging center while simultaneously doing monkey physiology at Princeton since 2005. She has almost literally a full page of key scientific appointments, and I will note only a few, including she's on the Scientific Review Board for the Howard Hughes Medical Institute, a major funding organization. Whoops. Ooh, that's nice. Sorry, should I? Thank you. It's the Howard Hughes did it to me. Editorial board, multiple editorial boards, chief editor of numerous journals, external science advisor to multiple countries, including Switzerland, Germany, India, and Israel. And most recently, she assumed the role of editor-in-chief of the Journal of Neuroscience, one of our major journals historically, and just a very important journal. She's also served as chief editor for neuroscience for Frontiers for Young Minds, and this gets to me into the humanity part. She's done that since 2015 and has been instrumental to the global growth of this endeavor to take science to young middle school kids of all socioeconomic statuses across the globe. Again, notably, assuming the chief editor position at J Neuroscience requires abandoning other editorial commitments. But I am incredibly grateful that she basically told the Journal of Neuroscience no. And she declined to resign from the Kids Journal and continues as the chief editor for neuroscience for the Kids Journal, in addition to being the chief editor for Journal of Neuroscience. So thank you very much, Sabine. In recognition of her contributions to the education of kids across the world, she received the Society for Neuroscience Award for Education in Neuroscience in 2019. I think this highlights her amazing level of commitment to not only science, but the betterment of humanity. As you might guess, She has received numerous honors, including election as a member of the American Academy of Arts and Sciences, and election to the National Academy of Sciences in Germany, to name a few. Again, there's lots of them. Her George Miller Award nicely closes her Cognitive Neuroscience Award sweep, since in 2005, she was also a recipient of the Young Investigator Award from the Cognitive Neuroscience Society. Congratulations to both. I will leave science to her widely anticipated George Miller Award lecture and only briefly mention a few of her many accomplishments, many centered on how we allocate attention to the world. The breadth and depth of her scientific acumen distinguishes her from many other cognitive neuroscientists. Most of us, including me, are very happy to master one preparation for study, whether it be human, monkey, mouse, etc. Remarkably, Sabine has mastered both human and monkey physiology, allowing her to provide seminal contributions to the understanding of behavior across both species. Really amazing. Her monkey work on the role of the pulvinar in control of cortical processing challenged the widely held dogma of a passive role of this massive thalamic structure. It was just a relay structure. She clearly showed that it was active in cognition and was very instrumental in controlling cortical activity. And as one might predict, given her dual role in human and animal research, she is now extending this work to human thalamic recordings in a recently awarded very large multi-center grant that she's the PI on. Her thalamic work is further enhanced by her findings of top-down fronto-parietal thalamic modulation, and her more recent work has revealed the rhythmic nature of attention. Again, upending the dogma of a static attention filter that moves across the field. What she showed, notably, she's provided conclusive behavioral and physiological evidence for rhythmic 4 Hertz attention sampling of the world in both monkeys and humans, doing monkey and human intracranial physiology. Quite remarkable. So how can she accomplish this fluid ability to span multiple species so elegantly? It's simple. Become an expert in neuroimaging, become an expert in behavior, become an expert in electrophysiology, including single unit analysis and local field potentials across species. Not the easiest task, but remember her postdoctoral training presciently set her up to be able to do this kind of really fundamental research. So, in conclusion, we have the trifecta. We have science, collegiality, and humanity in our 2023 George A. Miller Awardee, Professor Sabine Kastner. So before you start the anticipated lecture, I would like to award to you, says it's presented to Sabine Kastner, MD, PhD, the recipient of the George A. Miller Award, a recognition of your outstanding contributions to cognitive neuroscience, San Francisco, California, March 2023. Congratulations. Thank you.
Thank you. Thank you and good afternoon. It's, for me, an incredible moment for my career, but if you get introduced by such a good friend, that makes it just incredibly special. Thank you. Thank you for this wonderful introduction, Bob. So I would like to thank the Award Selection Committee and the Cognitive Neuroscience Society for this incredible honor. Indeed, my colleagues at Princeton are particularly proud of me that I'm bringing the Miller Award back to George's hometown. I actually met George Miller only 2 or 3 times, and I believe the first time was in 2005 when I received the Young Investigator Award. And George took me aside and he said, "I read your papers, and I think your paper has some potential." I hope that I lived up to George's expectation. Now, I'd like to dedicate this award to all the wonderful colleagues who have contributed to the body of work that we have generated during the last 25 plus years and that I'm going to discuss with you today in part. So let me introduce you to my GAM band. We modified this well-known album cover. It is at least well-known for people in my generation. As I just found out last week, this is not necessarily true for the younger generation. So if you do not know this album cover, don't worry, this is the original. It is the Beatles' Sgt. Pepper's album. And so what we did here was we modified this cover or this picture to contain all my students, mentees, and collaborators from the last 25 years. Every single character from the original Sgt. Pepper album has been replaced with a scientist band member in this version. I feel so enormously grateful that I had and continue to have the privilege to work with such an extraordinary group of talented scientists, many of whom became close friends. If you ask me what the essence of my scientific career is, it's this band. So I'd like to thank every single one of my band members for their inspiration, curiosity, dedication, and hard work. Special thanks go to my mentors. You see them over here. You see the cursor, hopefully. So these used to be the early Beatles. These are my mentors, Otto Kreutzfeldt, Leslie Ungerleider, Bob Desimone, and Ann Treisman. And another special thanks goes to my family. I think particularly female scientists depend on the support of their families, and my family has been my bedrock. And I would like to thank Sarah, Ben, and Michael. Now, let me also use this lovely band to introduce you to the topic of today's lecture. Everyone knows what attention is on its neural basis in the primate brain. The problem that has interested me for most of my career is how we select information from cluttered scenes like this. And one way to do that is to recruit a spatial selection mechanism that we often refer to as a focus of attention or a spotlight of attention in our textbooks. So the characteristics of the spatial selection mechanism is that it operates only over a small part of the visual space in front of us. And everything that falls into this focus is then processed in preferential ways through a network that I will introduce to you later. So what that does to me is it helps us to identify some of the band members here. So I introduce you to some: Diane Beck, Charlie Gross, Kian Nobre, Bob, of course, Marlene Berman, who is here too, Yuri Salman, and Michael Graziano. Now, you can move the spotlight of attention in dynamic ways across the scene to gather the information that you would like to identify band members in this particular example. And you can do that either in an overt way, which means you move your eyes along with the spotlight of attention. This is a relatively slow process and can happen every 250 milliseconds or so. Or you can do it in covert ways. That means you're fixing your gaze somewhere in that scene and then move the spotlight around without moving your gaze. And this is much more efficient and fast. It can happen every 50 or 60 milliseconds, so at a roughly 20 hertz rhythm. And I will tell you today some facts that we have learned, some knowledge that we have gained on this covert spatial selection mechanism that I will refer to as attention for the rest of this lecture. Now, we study these topics in two brain models, as Bob has already told you, the human and the macaque monkey brain. And we establish behavior, common behavior in both of these species. We use behavior as the glue to tie these two brain models together. So typically we will start from a very well-established tasks from human cognitive psychology, and then we try to establish similar behavioral patterns that have been established in humans in the macaque brain. What this gives us is to bring the results that we get on the neurophysiology, on the mechanistic level in the macaque brain much closer to the human brain. And I will give you examples for that today. Then we can combine the behavioral measures in these two brain models, a group of participants with neural signals, and we use a variety of signals that reflect my training in neuroimaging and intracranial electrophysiology. And the human intracranial electrophysiology that I will introduce you to later is done together with Bob and other collaborators. So let me start by just showing you what happens in the human brain during a covert spatial selection of the type that I've introduced to you with my GAM band. So let's hope for the best here, that the movie works, and miraculously it does. So what we're looking at here is a right hemisphere of the human brain. This is the posterior pole, so this is a cortical surface that we reconstructed, and we inflated it to make all the sulci visible. And this is the posterior pole here, and this is the frontal pole. And what we see are bold signals, functional magnetic resonance imaging signals that float through this brain. This is a visual task, so there's a buildup of activity over the occipital pole, the visual system proper, if you like, and then later a buildup of activity in higher order cortex along the interparietal sulcus that you see here, and there are also frontal sides that you cannot appreciate in this bird eye view, and there are subcortical network parts as well that, again, you cannot appreciate. But what I find amazing is that with just a very simple cognitive task like this, allocation of attention in space, you activate a large-scale network that spans all lobes of the primate brain and several other sources. And the network that routes the visual information that is selected through this network must be extremely efficient, because these kinds of selections happen on the time scale of about 200 to 300 milliseconds. And we are interested to learn what these single nodes in this network are doing and how they exchange information to make this process so enormously efficient. Now, very early on during my imaging career, we asked the question, is there a functional structure or organization in this network? And I want to start with a study that I did as a postdoc in Leslie Ungerleider's lab. Leslie had, as some of you know, an enormous influence on my career as a scientist, and she shaped my scientific thinking in unique ways. We will celebrate her life as a scientist in a special symposium on Tuesday afternoon that I would like to advertise, and I hope that many of you will still be there. We have a wonderful lineup of speakers to honor her. Now, the work that Peter de Wert, Bob Desimone, and Leslie and I did in this particular study was to reveal this kind of functional organization and this large-scale network for, I think it was one of the first demonstrations of this type. The experiment we did was very simple. It was a Posner-like attention study. So we had subjects direct attention to the periphery of the visual field, as schematically shown here with the spotlight of attention, but this was, of course, never on the screen. And this was then followed by a period when an area of visual stimuli was presented, and the subject had to select one of the four stimuli here out of this array. Now, what we saw in terms, so this was in human subjects in this imaging study, so what we saw in extrastriate cortex, also in striate cortex actually, was the following pattern. So we saw an increase of fMRI signals during that expectation period. So this is a period where there is no sensory input to the system whatsoever, which was then followed by a further increase once the attended stimuli were kicking in. So there was this two-stage pattern. At the time, when we made these observations, this was quite radical because the visual system was thought to be, like a bottom-up system. And this was a very clear demonstration that this system could be driven solely by cognitive signals, here by this top-down attention signal. Now, what we saw in higher order cortex was quite different. So this is from the human frontal eye fields. There we saw that there was a larger increase of activity during this expectation period, which was then plateaued. So there was no further significant modulation during the period with attended visual stimuli. So this activity pattern seemed to reflect the cognitive operations of the task, the allocation of attention in space, and as long as that happened, we saw this strong signal, and then it would get redirected, or during the redirection of attention, it would then fade out, basically. So this way, we could dissociate areas that were driven by cognitive signals from those that were kind of driven by both cognitive signals and visual signals, like extrastriate areas here. So also parietal areas would show this kind of plateau pattern. Now, from experiments like that, we very early on came up with this top-down model of attention where we have a fronto-parietal network that basically controls the feedback that goes into these visual representations and modulates the ongoing visual processing. And I would like to challenge this top-down model, or I want to challenge and extend it actually in my talk today by showing you that there are other forms of control than this top-down control and other control centers. And I will talk about the thalamic control that we have learned about during the last 10 plus years. So I want to introduce you to thalamic timekeeping and to a concept of distributed attention control. And the study that I would like to highlight is the early study that we did on this topic. So you see quite a few band members here that have contributed to this topic, but I will highlight the work of Yuri Salmon and Liang Wang. Now, this is a schematic here of the primate thalamus, and the visual thalamus consists of two nuclei. I think you all have heard about this one over here, that is the lateral geniculate nucleus. This is the nucleus that projects or relays the information from the sensory periphery, from the retina to the cortex. We were interested to study this very, very big next-door neighbor here, the pulvinar nucleus. This nucleus is, in contrast to the lateral geniculate nucleus, almost exclusively interconnected with cortex. It receives input from cortex and sends input to cortex. Now, if we look at the structure in terms of its anatomical connectivity a little bit more closely, we find a quite intriguing pattern. So these are the 6 layers of cortex here. This is the pulvinar in the center of the brain. And what holds as basically an anatomical rule in the primate brain is that any area that has a direct cortical-cortical connection, and I have here 2 areas along the ventral visual pathway, V4 and TO, for whom that is true. That is this connection over here that goes from the superficial layers of the lower area to the middle or input layers of the next area. So if 2 cortical areas have a direct projection that they share, then they also by default have an indirect pathway that goes through the pulvinar, and it originates from layer 5 and then goes into the superficial layers of the next area. So in this sense, you can think about the pulvinar as being made up of projection zones that interconnected cortical areas share. And given that there is such an intricate anatomy, we were wondering whether there's also function that goes with it. And so the experiment that we did at the time was to record from 2 interconnected cortical areas, and we used these areas here along the ventral visual pathway, V4 and TO, and then to visualize their projection zone in the thalamus, in the pulvinar, and record with a third electrode from there. So before I introduce you to our attention task, I would like to develop the specific hypotheses that we were testing here based on the anatomy that I tried to lay out. And this hypothesis was based on a number of facts that we had in our hands when we started this work. So one known was that in a cortical area, the occurrence of spiking activity is often observed at certain phases of oscillatory activity that you can measure in the population responses like the local field potential. So here in the schematic, the spiking activity or the most excitable window of activity for this population in this putative area here would occur at the peak. Now, if two areas want to exchange spiking activity, or any information that goes to spiking activity, then it is advantageous to synchronize this oscillatory activity that sets or controls the excitability of the neuronal population, so that a spike that travels from area 1 to area 2 meets an excitable window, like in this example, and not an inexcitable window, like in this example over here, when the spiking activity would meet a trough, so a relatively inexcitable window of the population coding here. Now, if we just kind of take this very simplified model of interareal interactions and think about it a step further, then the question is, how do these two areas now negotiate what a good state of communication is? And one way to do that is, of course, to use the feed-forward and feedback pathways that they may share. But an alternative and mutually not exclusive way would be to involve a third area to do that on their behalf. And we thought, of course, the pulvinar could play such a role. This is computationally probably the more elegant solution for a network, because the pulvinar could not only do that for two areas, but for a large amount of areas, so for a large-scale network, basically. So the hypothesis that we probed in these studies was that the pulvinar may synchronize neuroactivity in two interconnected cortical areas in order to optimize information transmission between them. Now, the signals that we were probing or analyzing in these studies were local field potentials. This is an example here of a raw potential. And what we can do with these signals is we can filter them into the frequency domain and then look at band-limited effects and also how those get modulated by attention. Now, let me introduce you to the spatial attention task that we probed in these studies. It was a variant of an Erickson flanker task. The task goes like that. There's a cue that directs attention either to this location, would be here the receptive field in this population that we are recording from, or to the opposite location. So this would be the control condition, basically. Then there is a delay period during which there is no further sensory input. It's basically a relatively stable cognitive state that we are probing. And then we show a target array made out of bow tie and barrel stimuli, and the animal tells us with a lever response, so there's never an eye movement in these studies, whether there is a bow tie or a barrel in the cued location. So the period that we are particularly interested in is this delay period when the attention network gets set up for action. We wanted to know whether there is a particular frequency at which the cortical areas would synchronize. And so we looked at that. This is a coherence measure between before and TEO, a measure of neurosynchrony across these areas. And you see that they set up here synchrony that gets modulated by attention, so that would be the red line as compared to the blue line, which is the unattended condition or attend-away condition. And you see that the modulation happens in a frequency band around 10 hertz. We often refer to that in the EEG literature as an alpha frequency band. And the same frequency band gets also modulated in the interactions of pulvinar and these cortical areas, so we see that across this network. Now, what this tells us is that this network uses an alpha frequency band during that delay period when the attention network gets set up, but it doesn't really tell us much about these two kind of communication models that we are probing here. And so we wanted to push this a step further by conducting a Granger causality analysis. A Granger analysis is based on the precedence of neuroactivity patterns in one area that can be observed earlier than in a second area. And so if we observe that, we assume that the area that shows this pattern earlier is the one that passes it on. And so if we look at the data in this way, what we see is that the pulvinar has an impact onto V4, a Granger causal impact. So it seems to impact the cortical area, also TO. I don't show you the result here, but it looks very similar. And again, this Granger causality is modulated by attention. So that would support this kind of model where the pulvinar is basically synchronizing these cortical areas during the delay period. Now, but what are the cortical areas doing, if anything? To our surprise, they do not seem to communicate during the delay period, at least not by this measure. So this kind of alternative model here is not really what we observe. So as you can see, there's no modulation whatsoever. So we think that the pulvinar is setting up this cortical attention network, and this is not only true for these two areas that I have highlighted in this study. We have seen that in many follow-up studies for other parts of the network. So here you see this study that included V4 and TEO. We have also observed that in a study where we looked at the interactions of the pulvinar with V4 and posterior parietal cortex, the lateral interparietal area, and also in between higher order areas, so the lateral interparietal area and the frontal eye field. So we think that it's a global mechanism across this network that holds wherever we have looked thus far. So let me summarize by saying that the pulvinar regulates synchrony between cortical areas during spatial attention, and we think that the pulvinar may play a function as a timekeeper, so controlling the temporal dynamics of cortical large-scale networks and synchronizing areas, aligning them in time, basically. So this extends our standard model of attention control. We think that in going forward, we need to look at different types of controls. So here you see the control of time. We have to ask, basically, we have to go away from these static top-down models. We have to ask whether there are other forms and other areas that are engaged in the control process. And here I've given you one example. Now, let me go to the next part of my talk and go back to the GAM band. When I introduced the spotlight of attention to you earlier, I told you that we can move the spotlight across the visual scene dynamically to get the information that we are interested in. But once the spotlight is allocated, what we seem to perceive is that we take in that information continuously. And I want to show you today that that is just a really big illusion. What really happens is that the spotlight goes through phases of better intake of information and worse intake of information. And that has behavioral consequences. So the spotlight is basically, once it's allocated, blinking, like in this animation here. So let me introduce you to the rhythmic properties of selective attention that give us, I think, very unique insight into the dynamics of this process. So a large band has been involved with this research, including Bob. And I want to highlight particularly the work of Ian Fiebelkorn and Randolph Helfrich today. So the task that we probed in these studies is a task that was actually developed here at UC Berkeley in the early 90s. It's an Egly-Driver task. The way the Egly-Driver task works is that participant is catching fixation, then we present these bar-shaped objects here to the right and the left of fixation that can be vertically or horizontally laid out. Then we spatially cue one of the four corners of these objects. So here it would be the upper right. Then there is a variable delay period, and then we present a target with high probability in the cued location. And that target is presented at threshold, so it's much dimmer than in this schematic here. This is a very hard task for the subjects. Now, what we were particularly interested in is what happens during this cue target interval. Is attention function just kind of steady over time, or does it show something more interesting? And this is a study in human participants. And as you see right away, if we plot the success rate as a function of the cue target interval, there are rhythmic fluctuations of performance here. So if you were to do this task right now, I could tell you right away when you're good at it and when you're not. So for instance, at this point in time here at about 550 milliseconds into the cue target interval, you are about 5% better to make detections, and that is at threshold. This is a really large effect, actually, than about 120 milliseconds later, where you go through a trough and then you go back to a peak and so on. So there is a behavioral rhythm there that goes at about 4 to 6 hertz. There's some individual variability in that, but that is the range of that behavioral rhythm. Now, when we made these discoveries in humans, that was a moment when I really lost hope that we could probe this paradigm, the Egly-Driver task, in monkeys, because humans are snobs and they sometimes think that they're special. And I thought this must be a new edition of evolution to the human brain. Well, I was wrong. Here are results from monkeys, and our non-human primates show these exact same behavioral rhythms. They're even aligned in many ways in the temporal structure. So now we are looking at a rhythmic property that goes back more than 20 million years in evolution. And that made actually these rhythms even more interesting. So let me just summarize the behavioral data by telling you that behavioral rhythms at 4 to 6 hertz occur during visual spatial attention, and they set up alternate states of better or worse performance that we can measure behaviorally. And these rhythmic properties of attention are preserved throughout primate evolution. Now on to the neural basis of these rhythmic properties. I will start by telling you about some results in our own species that we obtained with intracranial physiology in epilepsy patients. So we did a series of studies in epilepsy patients using electrocorticography. So here are two examples of grids that are implanted in the brains of these patients. This is to prepare them for their upcoming neurosurgery to remove the epileptic tissue. So there is no scientific purpose here. This is done for clinical diagnostic purposes. And given that, the kind of coverage that you get in each patient is variable. So here are 4 examples. These red dots denote the electrode locations rendered on the patient's brain. When we put together a number of these data sets, so typically 8 to 10 or so, we get actually beautiful coverage over typically the language-dominant hemisphere, so the left hemisphere. This is one reconstruction here from the study. So here you see all these electrode recording sites in a lateral view, in a medial view, and in a posterior view. And you're left with something like 1,000+ electrodes, which is really amazing. And now what you can do to kind of elucidate a little bit what the underlying functional properties of the tissue that you're recording from is, you can combine these electrode sites with atlases, and we use here an atlas of the human visual system. So here you see color-coded all these visual areas that people have described in the field. This is based on our own data from the lab, and it's a freely available atlas that can come in very handy. So when I refer to specific sites in the human recordings, this is how we reconstructed them. So what we are looking at in these recordings is, again, a field potential. But this field potential is much coarser than the one I've showed you from the monkey data. So this is fed by thousands and thousands of underlying neurons. But we do basically very similar analyses, so we filter that in the frequency domain. And the signal that I will focus on is a high-frequency broadband signal. That is a signal above 70 hertz or so. This is a very complex signal, but the way you can conceptually think about it for the purpose of this lecture is as multi-unit activity, just fed by a lot of neurons. And this signal is so powerful that you can even look at single trials, and that is what we did in our patients. So here's, again, the schematic of our Egly-Driver task that they are performing. And here is a single trial that was recorded in one of the posterior parietal cortex attention network sites. Now, what we see here is the cue-evoked response at this point. This is the beginning of the cue-target interval. This is where the cue-target interval ends. The broken line here is the time at which the target at threshold occurs. And then this green line here is the manual response of the patient, so the detection of the target. Now, what you can see right away is that during that cue-target interval, the high-frequency broadband response is not flat, but it is modulated in a rhythmic way, so it goes to these fluctuations here. And I want to show you that this is not the only trial we ever found in this data set, but this was a very regular finding. Here are two more trials from the same site and the same patient. We see it even in a case like that, where the patient actually misses the target. So the first question that we had was, what is the frequency at which this modulation of the high-frequency broadband response happens? And to get at that, we did a peak-triggered average. So this analysis basically takes a peak, and it takes a time window before that peak, and after that peak, moves it over to the next peak, and so on, and that way averages a lot of data, and so on. So for this site, when you do that, and it's done here on the raw potential that you see in black, we took a 500 millisecond before the peak and the 500 millisecond after. We fitted that with a sinusoid. You see very clearly that that modulatory rhythm is a 4 hertz or a theta rhythm. So now we have a 4 hertz behavioral rhythm, and we have a 4 hertz neuronal rhythm. But that doesn't tell us that these rhythms have anything to do with one another. So this could be a pure coincidence. So we wanted to see whether there is a way to link the neuronal signal and the behavioral outcome in closer ways. I want to go back to these single trials to give you the kind of intuitive idea of this analysis. So we looked at the phase of this theta 4 hertz rhythm that modulates the high-frequency broadband response in relation to the occurrence of the target. So here in this example, in this trial, you see that the occurrence is very close to a peak. This is also true in this example here. But in this example, when a patient is actually missing the target, this coincides with a trough of that modulatory frequency. So we asked basically the question, is the phase of that theta rhythm predictive of behavioral outcome? And I wouldn't tell you about this extensively if it wasn't true, obviously. So when we do this analysis here, you can see very clearly, this is the theta phase, this is the hit rate, that there is a phase in this theta rhythm that is associated with a poor performance, and then another phase that is associated with much better performance. We call these phase detection relationships, and it gives us a way to tie together a neuronal signal that we can measure at the level of a large-scale network that may be very important for aerial interactions and link it to behavioral outcome. So we looked where we would find these kind of phase detection relationships most prominently, and we find them in fronto-parietal cortex. So all the hot colors here in this patient's brain over here, and frontal cortex and over here in posterior parietal cortex. Those are the regions and the sites we find with these very strong phase detection relationships. So this is not like a random topography, but a topography that involves this fronto-parietal network. Now, let me briefly summarize what I told you about the human data. I've told you that high-frequency broadband power is modulated by theta oscillations across a fronto-parietal network, and target detectability depends on theta phase. Now, during the last part of my talk, I want to give you an example for how far we can push mechanistic insight if we involve a non-human primate model. So I want to tell you about some of the results that we obtained in our non-human primates who also show these attention rhythms, as I've shown you earlier. Now, as I just told you, the human work had basically pointed us very strongly to fronto-parietal cortex playing a very important role in these behaviors, and so we decided to record from two areas that have been implicated in non-human primates extensively with attention function, and that is the frontal eye fields in frontal cortex and the lateral interparietal area in posterior parietal cortex. Now, the way I depicted the fronto-parietal attention network thus far is as some kind of control network that's producing signals that get fed back into visual representation areas. In this part of my talk, I actually want to change that concept a little bit, and I want to tell you a little bit about the neural response properties that we see there, and that we can also have other concepts about this fronto-parietal cortex. Now, the neurons that we typically see there fall into two categories. One category are visual processing neurons. So here's an example. Actually, the checkerboard will be my visual icon for the rest of the talk. So this is a neuron that responds strongly to a visual stimulus, but it does not respond during eye movements. So there's no response there during a saccadic eye movement whatsoever. And then you see the opposite response properties. So you see oculomotor or movement neurons that respond during saccadic eye movements, but not in response to visual information. So the way, and then you see, neurons that have a mix of these two properties. So the way I would like to conceptualize this fronto-parietal network is that it is a network where two main other networks or subnetworks feed in. One is the downstream visual processing system, the visual system proper, and the other one is the oculomotor system. Now, if you think about these two functions, they are potentially in conflict. If you want to take in information from the visual environment, you shouldn't move your eyes, and if you move your eyes, you can't take in information. So my argument will be that these theta rhythms that we see that drive these rhythmic properties of attention, that they are a way to coordinate and avoid this functional conflict that you have potentially in this fronto-parietal network. So let me show you some data to make my argument here. So the first thing we had to look at is whether there are phase-dependent relationships First of all, whether there is a theta rhythm in these fronto-parietal regions in non-human primates, and if so, whether we would also see these phase-dependent detection relationships. So we looked at that, and we saw that in both of these areas. So this is plotted a little bit differently than in the human data, but here you see theta phase, and you see the difference in hit rate in the frontal eye fields and the lateral interparietal area. And you can see very clearly that the theta phase predicts behavioral outcome. So you have a good theta phase, we call it a good theta phase, where your performance is about 5–6% better than during the alternate phase here in this region. And you see that in both areas. You see that also in other parts of this network. Now, we have the good and the poor theta phase. In this data set, since it's so much larger than what we typically sample in the human brain, we can now ask additional questions that we just simply cannot ask in a human data set. One is, are there higher frequencies that would also set up these phase detection relationships? And if so, are they associated with the good or the poor theta phase? So how do they relate to behavior? So we did that analysis, and what we found was that there's prominent beta activity in the frontal eye fields that is predictive of behavioral outcomes, so it sets up these phase detection relationships. But this beta activity is exclusively associated with the good theta phase, so it's predictive of good performance. And in the lateral interparietal area, we see faster activity that goes into the gamma band up to 45–50 hertz that is also predictive of behavior. But again, it's exclusively associated with the good theta phase, so it is predicting good performance and not poor performance. So let me try to tie this together in what we have thus far. So we have a theta rhythm in the frontal eye fields, that's this one here, and in the lateral interparietal area. This theta rhythm is phase synchronized. I haven't shown you the data, but you can take my word. And then we see during the so-called good theta phase that is associated with better performance, we see a prominent beta rhythm in the frontal eye fields and a gamma rhythm in the lateral interparietal area, and both of these rhythms are coupled, so controlled by that slower theta rhythm. Again, something you just have to take my word for. So what does it all mean? We got some really interesting insight when we went back to the single cell properties in these areas and how they may relate to this oscillatory activity that we can measure in the population. So when we sort these neurons into these visual and oculomotor neurons that I introduced to you earlier, what we see is that the spiking activity of visual neurons in the lateral interparietal area is locally coupled to gamma activity, and it is also coupled to gamma activity in the population signal that we measure in the frontal eye field. So we think that this activity from these visual neurons uses this gamma channel to propagate that activity into the frontal eye fields. Now, this is not so surprising because gamma activity has been associated with visual processing in the visual system over and over, so this association of visual neurons and gamma activity is quite consistent. Now, what we saw in the spiking activity of neurons in the frontal eye fields is that oculomotor neurons were correlated with beta activity. And that is true locally, and that is also true for the population activity that we measure in the lateral interparietal area. So we think, you can think about this as a feedback signal that these oculomotor neurons send into the lateral interparietal area. We think about it more broadly as a signal that is associated with the suppression of movement. That is what beta activity typically is in the motor system. So if we take this information that uses the single unit properties and the spiking activity in relation to these population measures together, then what we are looking at is the following dynamic here. We think that during the good theta phase, we have the visual neurons associated with the gamma activity that promote the visual processing into higher cortical areas. So it's promoting sampling. And at the same time, we get a suppression of the oculomotor system to avoid eye movement. So this is the coordination of these two functions here. And this is then alternated with a state where you have a relative release of suppression from the motor system and a relative suppression of visual processing. And we think what could happen during that state is actually that you shift attention to a new location. So this state here, the so-called poor theta phase, is worse in terms of visual detection behavior, but it may be preferred in terms of motor behavior, and that is something that we are probing right now in the lab to substantiate this idea. So then you may think, why the heck is this setup so complicated? Couldn't it be a little bit easier? So we had a lot of problems in the beginning to convince our peers that these ideas may have some merit here. And I think we can get a clue why this is such an advantageous temporal dynamic by looking at clinical populations. So if you look at attention deficit disorder, what we see in those kids, it's a huge spectrum, but at the extremes of the spectrum, we see kids that are hyper-focused. I think about them as being basically locked into the sampling states, so the good state over here. So these kids they do not have, properly speaking, an attention deficit because they can play chess for hours. But you will never get them to the dinner table. So their problem is that they cannot switch. And then on the opposite end of that spectrum, you have kids who go from activity to activity. They're hyperactive. So they seem to be locked into this kind of shifting state. But what these kids may not be able to do is to set up these rhythms that would afford them to do the sampling and the shifting of attention on the fly, basically, because they have that opportunity every every 250 milliseconds, 4 times per second. You may not choose to, switch attention so frequently, then you can go back to sampling, but you have that for free, basically, with this kind of dynamic that we reveal here in these rhythmic properties of attention. And to probe ADHD kids and other populations with attention deficits is another emphasis that we have right now in the lab to push some of these ideas to the next stage. Now, I have shown you in this last part that attention function unfolds over time and has rhythmic properties. Theta oscillations organize sensory and oculomotor networks into 2 alternating states for sampling sensory information versus shifting attention. And we think that the attention rhythms serve as a mechanism to avoid functional conflicts. So I have reviewed today in my lecture some of the knowledge that we have gained about attention function over the last 20 years or so. Our understanding of its neurobasis is still in its very beginning stages, I think. What I've shown you today is that attention function is supported by a functionally specialized large-scale network. Attentional control is distributed and includes thalamic centers, and rhythmic properties may account for the flexibility of the selection process. And with that, I would like to thank you for your long attention span.